And now for our final undergraduate finalist presentation. Please welcome to the stage Vilnius, Lithuania.
How are your feelings today?
Hello everyone, we are Team Vilnius, Lithuania, and this year we are very excited to present you our project Exullose. So let's start at the beginning. Biopolymers such as starch, protein, DNA, and cellulose dominate our surroundings as building blocks of nature and are utilized as food, clothing, and construction materials by humans. In recent years, researchers started to notice detrimental effects of fuel-based products and started to look for natural alternatives. However, this quest is not as easy as it seems. Biopolymers often lack some kind of properties that are needed for the modern world. For instance, bacterial cellulose produced by this non-model organism, Komagataeibacter xylinus, is known for its mechanical properties, for water retention. It is different from plant cellulose because of its homogeneity and because it does not have neither lignin or hemicellulose, but it do not contain any antimicrobial activity. It is hardly degraded in mammal tissues, and it's very hard to functionalize without the use of harsh chemicals because of its crystallinity. Therefore, we present to you our project Exullose, which is aiming to modify bacterial cellulose in vivo. The first way that we modified bacterial cellulose was a production of bacterial cellulose and chitosan copolymer, providing bacterial cellulose easily modifiable amino groups. The second part of our project is self-dyed bacterial cellulose that can be later applied in textile industry. And the third and final part of our project is bacterial cellulose and polyhydroxybutyrate composite, which is a plastic-like material. You may be wondering how they are connected. So they are all connected with the same part collection, the same synthetic biology tools, so the same plasmidic backbone, promoter, and terminator ribosome binding sites can create these three very different polymers. Because we were successful in completing all the parts of our project, so let's start with bacterial cellulose and chitosan copolymer. So because the enzyme that produces bacterial cellulose cannot recognize uridylated glucosamine, it can only recognize uridylated N-acetylglucosamine, we had to start with bacterial cellulose and chitosan copolymer. The first construct, the first operon for its synthesis, as you can see in the upper part of the slide, is made out of 3 chitosan metabolism genes from an yeast Candida albicans. When transformed into bacteria that synthesizes bacterial cellulose, this construct needs addition of N-acetylglucosamine into the growth media for its incorporation into bacterial cellulose structure. However, N-acetylglucosamine is really expensive. In order to make this polymer suitable for mass production, we created a second operon that you can see at the bottom of the slide. It is made of genes from alternative chitosan metabolism genetic pathway in yeast Candida albicans, and it enables bacteria to produce N-acetylglucosamine itself and also incorporate it into bacterial cellulose structure. So the first sign of our engineering success was this very cute blob that has like a looser structure compared to natural bacterial cellulose. we came to a conclusion that this probably caused by N-acetylglucosamine incorporation, because N-acetylglucosamine incorporation inhibits hydrogen bonding between the cellulose fibers. And, okay, oh, sorry for that. And later on, we found out that we were right. After fluorescent staining with wheat-germ agglutinin conjugated with Alexa fluorescent dye, we saw that N-acetylglucosamine incorporation was successful because wheat-germ agglutinin only binded to N-acetylglucosamine, in this case present only in the products of our operants that we constructed. The second part of bacterial cellulose and chitosan production was the deacetylation of bacterial cellulose and chitin copolymer. In this case, we used 3 different recombinant chitin deacetylases with broad substrate spectrum because this kind of polymer do not exist in nature. We also fused these chitin deacetylases with cellulose binding domain in order to increase the degree of deacetylation. And we were also successful. As you can see from this little cute peak right here, that is only present in our deacetylated polymers and corresponds to a mid-2 vibration. There were 3 amino groups in our polymer, and it proved our concept. So after all of this, you are probably wondering why we went through all this pain and suffering to create these polymers. So incorporation of N-acetylglucosamine into bacterial cellulose and chitin copolymer and glucosamine in cellulose and chitosan copolymer makes these polymers susceptible to lysozyme. And it makes them like biodegradable in mammalian tissues, which is really a good way to apply these polymers in biomedicine. Furthermore, the uncovering of amino groups in bacterial cellulose and chitosan copolymer gives the antimicrobial activity that bacterial cellulose was lacking. And also, these 3 amino groups are easily modifiable. You can just choose whatever molecule, almost whatever molecule, and just add to this polymer. And in this way, you can increase wound healing properties or any properties that you want in this polymer and widen its applications in biomedicine.
So a second part for validating our engineering success using synthetic biology for modification of bacterial cellulose consisted of creating self-dyed bacterial cellulose. At first, we evaluated 6 novel monooxygenases to see which works the best, and we selected the one that is shown in the construct. So we transformed this monooxygenase into xylinus, and we were able to produce a system that can make colorful cellulose. So not only the cellulose is colorful, but it is of different colors. As you can see, depending on the substrate that is depicted as the chemical formula here, put in the growth media, you can regulate the color you want. So we were able to achieve the colors ranging from blue to pink to brown. So you may be wondering why we wanted to do this. So this is an environmentally friendly way to produce textiles, because in textile industry, the majority of pollution comes from wastewater that is created during dyeing process. So if you entirely skip the step of dyeing the material and you grow the material already colorful, you eliminate you offset the pollution that is created by textile industry. As well, bacterial cellulose can be used as an alternative to leather since it has good mechanical properties. So the last part of our wet lab project that verified the modification of bacterial cellulose was creation of bacterial cellulose and polyhydroxybutyrate composite. So this plastic-like material was achieved by transforming cake xylinus with polyhydroxybutyrate synthesis operant. What is interesting and exciting about this part, that we were able to create not only constitutive expression, but also regulated expression of PHB synthesis. So this was important that because we could regulate the PHB content in our polymer. So firstly, we verified that the constitutive expression works by nail red staining that can only bind to PHB granules that are contained in colonies that have working PHB synthesis construct. So as you can see, there are a few orange, like red colonies that can be visible as compared to negative control of unmodified bacterial cellulose. And lastly, we wanted to show you the visual differences of this bioplastic material. So as you can see from the pictures, the pellicles are more connected. It looks like a plastic. And this was verified by scanning electron microscopy. As you can see, the mesh pattern is not as pronounced in the composite. The fibrils are more connected. The surface is more smooth. So this can be explained by PHB incorporation into the polymer structure. So how we can use this and how we can apply this? Since it combines attractive properties of both bacterial cellulose and polyhydroxybutyrates, you can create a plastic-like material that can be used, for example, for food packaging or other alternatives to petroleum-based products. And what is good about the system is that purification is quite easy, because you only need to boil the polymer that you produced to attain the bioplastic, therefore eliminating one of the key problems with applying PHB in the industry. Sorry. And I just wanted to conclude everything that this was connected with our part collection. And if you want to be a polymer designer as we are, you can mix and match different synthesis operants and create different bacterial cellulose composition.
Well, great. So, our wet lab clearly and undoubtedly achieved engineering success. But what we did for public engagement initiatives? And well, the answer is quite a lot. So, when we were brainstorming what we want to do and what we want to organize, well, we decided that we will focus on interdisciplinary approaches, especially with art, and do all of this in an inclusive manner. So probably the most ambitious project we did was Art Science, a 9-event cycle organized by us that aimed to introduce the general public to the development of science through a prism of art. So as I mentioned, we organized 9 events in total, so you could see some moments from each of them. So we started with an art exhibition, then continued with Mafia-inspired gameplay, role-play activity, and, well, did some alchemy, like marked the experimental phase of science in an orientational game perspective, and continued with very important discussion, superstitions, about the skepticism of science and the importance of scientific communications also in academic institutions. All of this journey continued, and we ended at the point where we speculated what could be the future of science and synthetic biology if it's used in a good or bad ways. So it was like super interactive, let's say, for the audience. The second project we did is to make sure that our project is inclusive. We published an introduction to synthetic biology in Braille. So here we saw a very big problem. We went to talk with our stakeholders, the teachers that work with visually impaired and blind, and we saw that although genetic engineering is in Lithuanian high school curriculum, people that are blind do not have accessible literature in Lithuanian in Braille. Therefore, we gathered all the information and published this book that is currently available in every single one of Lithuanian audiosensory centers and educational centers that works with blind and visually impaired. Also, we decided that we will leave Vilnius, leave the mass population centers of Lithuania. This was because most of scientific communication events are focused in the dense population centers, and regions are probably like left behind. So we went to 6 cities in total and talked about genetic engineering and synthetic biology and a little bit about our project with mostly senior-dominated audience. And well, all of this was done in a dialogue manner, so we were very happy to hear the feedback that their perception of what is GMOs changed a little bit after our activity. The last project was Atrex.bio. I won't talk about this a lot because we have a very limited time, but it's a free online learning tool that was created with Lithuanian teachers that teach biology, and this was corresponding to a changing Lithuanian curriculum, without adjusted learning and teaching materials. So this project is a long-term contribution to Lithuanian education, and we will make sure that this project is continued after iGEM. So in total, our outreach scattered all over Lithuania and reached 25,000 people. That's literally 1 in 100 Lithuanians that we engaged throughout our public engagement. We are very proud about this number, and all of this would not be possible without our team, our PIs, you guys, and our sponsors. Thank you.
Thank you so much.